Okay, so we're going to be discussing in this second lecture for chapter 3, we're going to be looking at what they call appendages. Okay, these are going to be bacterial structures that are outside of the cell. Okay, these things, the flagella, the pili, the fimbria that we'll be discussing, the capsule, all of these are outside of the cell. They are outside of the cytoplasmic membrane. This is going to be quite different than when we're discussing in chapter 4 eukaryotic cells. First up, we're going to be discussing flagella. Flagella are a means of locomotion, a means of motility. Motility, locomotion, just means that it can move from point A to somewhere else. Now we need to keep in mind with these bacteria that these bacteria are single cell organisms that do not make decisions. They do not make choices. Okay? Flagella spins, bacteria moves. It's random where it goes. Flagella are going to have three components: a filament, a hook, and a basal body. This is completely different than the eukaryotic one that you've probably learned in a previous course. A filament, which will protrude out. It is a series of proteins polymerized together. Generally, the flagella is going to be much longer than the cell itself. The hook, and then the basal body. The basal body will be embedded from the cytoplasmic membrane through the cell wall and an outer membrane if it's a gram negative, or capsule or anything if it's a gram positive. You look over here on the right, here is a transmitting electron micrograph picture showing you the basal body in the beginning of the hook. This is an actual motor. It's driven by hydrogen flowing from the space here between the cell wall and the outer membrane, flowing back through across the cytoplasmic membrane and into the cell to be pumped out later. Flowing through, causing this to spin. This spins, causes the hook to spin, and then the filament whips around. Think back to you're a little kid and you got your first jump rope, whether it's at home or at school. What's the first thing all kids do? They grab one end and they just kind of whip it around. It's exactly what's happening here. That filament whips around wildly, causing the bacteria to either be pushed or pulled through the liquid. Not all bacteria have flagella, so it's a yes or a no whether or not flagella are present. And if it's a yes, they're present, how many? Where are they found? Is it a single monotrichous, just one flagella? Lophotrichous, is it many but they're clumped together at one pole? Amphitrichous, are they clumped at both ends of the cell? Or is it just random wherever they are? Peritrichous. Now, I wasn't joking when I said that the flagella just kind of whips around. Now, granted, it's in water, so it's not going to be as wild as it was when you were a little kid. But this will push or pull the bacterial cell. They call this a run. It moves, and then the flagella motor stops, and the bacteria just kind of floats around, changes its orientation, floats around in three-dimensional space, whatever this water is. It works the same whether it's a single or multiple flagella. Here, the single can whip around wildly. If it's multiple flagella, it's going to have a little tighter control by the bacterial cell so that these don't get wrapped around each other. But it's still the same thing. The flagella spins, causes the cell to move. Flagella stop, and then it just kind of floats and tumbles around. That's where they get this idea of calling it the run and tumble. The run and tumble can be Basically random. Okay? It's a single cell organism. It's not choosing to go one way or another. It runs, it tumbles, and then it clicks on a run. Tumbles, spins around, and then runs. Tumbles, spinning around, runs. So you see here, they tracked it around and it's almost back where it started. But sometimes there's a signal. And it can be more of a particular movement. Now, other external structures can be fimbria. These that we see here, these green protrusions off the bacterial cell, are bristles. Now, it looks like it's just a ring, but you got to remember, this is a microscope picture, so these fimbria are all around it, all 3 dimensions. They surround it. They're polymers of proteins, long strands of proteins, glommed together, forming these bristle-like structures. They're great for adherence. Counter that to the pilus, which is plural, individual pili. This is a hollow tube, a long hollow tube. Donor cell up here, recipient cell here. They both have a pilus, a pili, sticking out. As the pili come into contact, donor will send over a chunk of DNA. Donor has an ability, whether it's drug resistance, ability to make a toxin, an enzyme. That chunk of DNA will be sent through the pilus into the recipient. The recipient will now have that ability. The recipient can then go and donate to another recipient cell. It becomes a donor itself. This quote-unquote mating is known as conjugation. It's how the killer E. coli that you hear for like Jack in the Box and a few other places eating bad hamburgers. E. coli 0157H7 has a toxin from Shigella dysentery, the toxin known as Shiga toxin. Somehow, many, many years ago, who knows when, Shigella formed a conjugation tube, a pilus, with an E. coli, and it swapped over. Now E. coli is a bloody killer. Other external structures, such as the S layer and the glycocalyx, you can see here on this gram-positive. How do I know it's a gram-positive? Because it has one cytoplasmic membrane, peptidoglycan cell wall, and that is all. You have an S layer, and then over that, a glycocalyx protective layer. The S layer, it's a protective layer that's relatively new. We don't know much about it because it only comes out, bacteria only produce it when they're in a hostile environment. Well, that's not going to be a laboratory environment. We're babying these bacteria, putting them in super rich broths and things to make it as easy as possible for them. So that's why this is something new. The glycocalyx we've known been around for a long time. The glycocalyx is a bunch of either polysaccharides or glycoproteins forming a coat around the bacterial cell. Now, those are 2 big words. Polysaccharides, glycoprotein. Poly meaning many, saccharide sugar. So these are long chains of sugars, hundreds of thousands of glucoses or other sugars strung together. Glycoproteins. Glyco means sugar, proteins. So these are proteins that have sugars added onto their surface. The glycocalyx is great for protection and adherence. Protection has this outer coating. It's like can either be a shell or it can be a slime layer. The shell known as a capsule. Slime layer, the polysaccharides and the glycoproteins are hydrophilic. They take up and hold water, so it makes it slimy. Strep pneumonia has a slime layer. By having the slime layer around it, antibiotics can't get to the cell. That's why once somebody gets a strep pneumonia infection, it's hard to fight it because it's hard to get the antibiotics to the bacterial cell through that slime layer. What we see happens is that some first cells that have a glycocalyx will adhere to something. This surface could be your teeth. It could be the valves in your heart. It could be a catheter. First ones can bind. Their glycocalyx now offers them protection and offers sites for other bacteria with glycocalyces to adhere to. And as more and more bacteria adhere, their glycocalyces kind of fuse, and then you get this big biofilm, this overgrowth on your teeth and your heart valve, on a catheter, different things.